ರೂಪಾ ಇಷ್ಟೇ ಇಷ್ಟು ಕೊಡು ತುಂಬಾ ಬೇಡ
Good evening, everyone. Anand, are you online?
Yes, sir. Am I audible, sir?
Yeah.
Yes.
But Anand, over to you. Thank you so much.
Okay. Thank you, sir. Good evening, everyone. Now we are 75 participants in this group. Shall we wait for 2 more minutes? Satyanarayana, sir, shall we wait for 2 more minutes?
Yeah, definitely.
Okay, sir.
Rudresh, welcome to the session.
Yeah, I request all the participants to yes. I request all the participants to mute themselves. More participants are joining. Now we are 91, 92. Once we cross 100, we'll start.
I guess it's a matter of a few moments. 99.
Last one to go.
One to go.
Okay, let us start. Good evening, everyone. Again, I am Dr. Anand Janagon, and it's my pleasure to welcome you all to today's masterclass. We are about to discuss a topic that is as old as antibiotics themselves, yet more critical than ever, that is beta-lactams. These were among the very first antibacterials discovered, widely used, and unfortunately often misused, leading to widespread resistance from bacteria. That's why today's webinar is so crucial. The topic for today's webinar masterclass is resistance to beta-lactams in gram-negative bacteria, mechanisms and detection methods. I am really excited to introduce our distinguished guest speaker, Dr. Rudresh S.M. He is a native of Chitradurga. He is currently serving as an additional professor of microbiology at AIMS BB Nagar, Telangana. He has completed MBBS from Mysore Medical College, MD Microbiology from Bangalore Medical College, and he has also completed DNB. Dr. Rudresh is a passionate microbiologist with keen interest in research. His areas of interest are antimicrobial resistance, molecular diagnostics, sexually transmitted diseases. He is an expert in molecular techniques like developing and validating PCR reactions, LAMP, cloning, protein expression, and purification techniques, to name a few. He has good experience in developing rapid diagnostics for infectious diseases and also carbapenemases. He has completed more than 10 research projects, many funded by prestigious bodies like RGHS ICMR and ICAR. He has more than 25 publications in PubMed Index journals. He has been an active and valued resource person in workshops and conferences organized by various organizations, including ICMR. So, let's welcome an inspiring microbiologist and an expert in this field, Dr. Rudresh. Over to you, Dr. Rudresh.
Thank you, sir. Thanks for the nice introduction. Sir, I'm audible, right?
Yeah, you are audible.
Yeah. I'm going to share the screen, sir.
Yeah, please.
Please check if it is okay. Hope, sir, the screen is visible.
No, yes. Yes, it's visible.
Yeah. And I'll just move it just to see whether it is working.
Yeah, it's moving.
Yes. Okay, sir. Okay. Thank you. So at the outset, I thank I am Casey for giving this opportunity to present the topic where I have worked for last 15 years. And I thank the president as well as all the organizers for giving this opportunity. So not wasting much time, let me take you through the topic. So today I'll be dealing with the resistance mechanisms of beta-lactam antibiotics among the gram-negative bacteria and their detection methods. So we know that the antimicrobial resistance has become a pandemic of 20th century, and it has crossed all its limits. We can say more deaths are occurring. It is the number 2 killer in the among the mortality if you see the global statistics. The disease is not only prevalent in India, it is worldwide pandemic, and most of the available antibiotics are under threat, and there is no new antibiotics which are available in the pipeline. So hence, it is our responsibility to understand the mechanism of resistance and also detection methods so that we can give an appropriate report to the clinicians on time. So this particular discussion will not go more of will go more of the technical part rather than the clinical part. So we can have the clinical part in the subsequent sessions if required. But right now, I'll take you through the core mechanisms and the methods in which they are detected. So the beta-lactam antibiotics, the term defines for a class of antibiotics which has a common tetra four-membered ring in its chemical structure. So we have penicillins, cephalosporins, carbapenems, and monobactams, which are the different classes, major classes of the antibiotics which we use in our regular practice. And we know that beta-lactam antibiotics are having very less side effects, and some of them, they have narrow spectrum as well as broad spectrum of activity. So based on that, the beta-lactam antibiotics have been used extensively in the clinical practice. So the classes, whatever I had told, though they were invented after 1940s, but the antibiotics are being produced by the major group of bacteria belonging to Streptomyces, Nocardia, and the some of the other minor groups. And most of the classes of antibiotics are naturally produced by this bacteria. And maybe we can consider this is an evolutionary mechanism which has taken millions of years to come down rather than occurring after 1940, where the Alexander Fleming invented the penicillin. So can we mute Trishya Das? I think some noise is coming. Yeah. So this is just an overview of the peptidoglycan synthesis, because we know that the beta-lactam antibiotics target the penicillin binding proteins, and these penicillin binding proteins are important in the terminal steps of the beta-lactam cell wall synthesis. Okay, so I'll not explain the first portion of the cell wall synthesis. Say once the cytoplasmic N-acetyl muramic acid and N-acetyl glucosamine is synthesized, with the help of a bactoprenol, the molecule will be shifted to the periplasmic space, and in this periplasmic space, the further interlinking of N-acetyl muramic acid, the peptidoglycans to the chain, will be done by transglycosylation, as well as the cross-linkings will be done by transpeptidation, and the left out amino acids of diala, which is terminal diala, what we see in the pentapeptide chains, that will be removed by the carboxypeptidases. So if you remember, there are two enzymes which are involved. That is, one is transpeptidases and carboxypeptidases. These two enzymes are called as the penicillin binding proteins. So we can classify these penicillin binding proteins based on molecular weight, like high molecular weight PBPs or low molecular weight PBPs. High molecular weights are one, as I told, like transpeptidases, carboxypeptidases, or endopeptidases, and low molecular ones are also same, but they are having a less than 50 kilodalton size. And gram-negative bacteria, they usually possess 7 to 10 penicillin binding proteins. So why we need to understand about penicillin binding proteins is different classes of antibiotics, and again different molecules have different affinity towards different penicillin binding protein. For example, penicillins, if you see, they have more affinity towards gram positive penicillin binding proteins, whereas aminopenicillins, they have again, they are more affinity towards gram positive penicillin binding proteins, and also they can bind to few of the gram negatives. So similarly, the other groups can majorly the carbonicillins, iridopenicillins, cephalosporins, monobactam, and carbapenems, they majorly bind to the main penicillin binding protein, what is called as PBP3. Okay, so we know their spectrum of action. This spectrum of action depends upon which PBP they bind, which beta-lactam bind to which PBP, and also how many PBPs they can bind. So this all depends upon the spectrum of action of a particular antibiotic. So this is the core structure of the beta-lactam antibiotics. You can see this is a 4-membered ring, which is called as beta-lactam ring. And if you can observe, this is present in all groups of the antibiotics. This is a penicillin, this is a cephalosporin, and this is a carbapenem, and this is monobactam. Okay. So what differs between all these molecules is the attached or fused 5-member or 6-membered rings. For example, if you see penicillin, it is having a 5-membered thiazolidine ring. Okay, but if you see the cephalosporins, it has a 6-membered dihydrothiazine rings. Okay, whereas carbapenems are having a 5-membered pyrroline rings, and monobactams, they don't have any rings. Not only this, they also possess the side chains, the R groups, what we have put in this picture. These R groups will make a new molecule, and based on the template of this penicillin or the cephalosporin molecules, further newer cephalosporins have been designed. You can see this is the R groups which is attached to the core background, and based on this R group, their affinity towards various carbapenemases will differ. Okay, so or various PBPs will differ. Okay, so Sidhendra sir, can you please mute? Surinder sir, can you please mute
PG
yourself?
master class. PG master class is going on.
So
okay.
let's come to the mechanism of beta-lactam resistance.
Beta-lactam
So
resistance.
I generally tend to start like intrinsic and acquired resistance always. And though beta-lactams do have intrinsic resistance, and acquired resistances has three major mechanisms. One is destruction of the beta-lactam antibiotics through beta-lactamases, decreased affinity of the particular penicillin binding proteins, or decreased concentration of the antibiotics, either with the loss of porins in terms of decreased entry, or throwing it out more by more and more efflux mechanisms. But there can be multiple mechanisms, which is a combination of, for example, a flux pump with beta-lactamase production or porin loss with the hyperproduction of NAMC. So combination mechanisms might be there, and multiple carbapenemase or multiple beta-lactamase production, which is more oftenly seen in these days. I have seen a maximum of 7 to 9 beta-lactamases in one particular isolate. And multi-drug resistance is again a threat, wherein the selection of these beta-lactam resistances has been still taking place. This is in one picture. We have depicted all the mechanisms of carbapenem resistance. Let's come to the intrinsic resistance. So I'll not go to individually organism by organism. This intrinsic resistance to few beta-lactam antibiotics by certain organisms, this information is essential for a clinical microbiologist while interpreting the AST pattern, especially by CLSI. If you just follow the CLSI, all these things will be omitted in their list of antibiotics to be tested against a particular pathogen. But by mistake, if these things are being tested also, you should not report, because they are intrinsically resistant for that particular organism. For example, I'm just giving an example of penicillin, ampicillin, or first and second generation cephalosporins cannot be used for Pseudomonas. Okay. Similarly, Acinetobacter, Enterobacter, Citrobacter, Serratia, Stenotrophomonas, Morganella proteus, Burkholderia, all these organisms, they carry the chromosomal AMC beta-lactamases, wherein if you try to give a beta-lactam antibiotic, the middle end products or the in-between products of the cell wall synthesis will stimulate the hyperproduction of AMC, that lead to the more and more resistance to these beta-lactam antibiotics. Hence, they should not be used for these particular organisms or different classes. Like particular classes should not be used for particular pathogen. This list should be depicted in your clinical microbiology laboratory so that you will not miss or report these things. Let's coming to the little bit of history of beta-lactamases. So since the introduction of penicillin in 1940, and in 1942 they started using in World War II, and by 1942 itself, some of the researchers, they reported the penicillinases in Escherichia coli K12 strain. Okay, so that was a cephalosporinase as such, but it was reported it's a CMY type, and this was hydrolyzing the penicillin. Hence, initially the penicillins were not used for gram-negative organism treatment, whereas they were mainly used for gram-positive organisms, especially Staphylococcus aureus infection. But by 1949, the penicillinase producing Staphylococcus aureus was identified. And later on, then the invention of new molecules is followed by the corresponding enzyme occurrence. So this is a latest article published by Karan Bush, where she has put the sequence in which the different beta-lactamases have been reported through after 1940. Does it mean that the penicillinases or beta-lactamases arise or they got origin in 1940? It is not so, because if you see, beta-lactamases are not only present in bacteria, they are present in fungi, they are present in us also. For example, the dihydropeptidase, which is present in the kidney, which hydrolyzes the imipenem. That is why we add on the cilastatin to prevent the increased hydrolysis of the imipenem in the kinetics. Okay, so if you see, many organisms, they possess the beta-lactamases, and beta-lactamases have a more history than 1940, that landmark of 1940. So some of the American researchers who explored the caves which were not or which were untouched for almost millions of years, for 3 or 4 billion of years. From there, they got the samples and they cultured bacteria. And to their surprise, they found that 60% of the bacteria they were possessing beta-lactamases. That means beta-lactamases has a long history, or their evolution is very long, maybe millions of years since the origin of bacteria. And so we as a human, where we entered in the nth moment, the 55th minute or 59th minute, we can say. So we too have certain beta-lactamases which can degrade the beta-lactam ring. So here, the origin of beta-lactamases is still a research wherein they have already they have predicted, based on the folding structures, the beta-stranded pleats or the alpha helixes. If you compare with the beta-lactamases and penicillin binding proteins, they have major similarities. For example, serine is an active site of beta-lactamase. Similarly, the penicillin binding proteins also, when they are binding or they are doing the transpeptidases or carboxypeptidation, they are going to use serine as an active site. And also, the conserving, like the conserved sequence motifs are present, though the amino acid sequences may be different, the conserved sequence motifs are present. And this will, and also the binding ability or catalytic domains, if you see the activity of the enzymes. So PBPs and beta-lactams, they share some of the properties. Okay. And so there is a conclusion that the beta-lactamases evolved through the various mutations, either duplications or divergence from the ancestral PBPs, and they have become, they have got the capacity to hydrolyze the beta-lactam. Why this beta-lactam hydrolysis and beta-lactamases? It is, as I told in the second slide itself, majority of the bacteria, they can produce beta-lactams. Okay, and for the survival sake, bacteria has adapted itself and started producing penicillinases or beta-lactamases, which can degrade those, and it gives a survival benefit for the bacteria. So now coming to the beta-lactamase per se. So beta-lactamases are the enzymes which inactivate the four-membered beta-lactam ring of the beta-lactam antibiotics. So as of 2025, if you see, 4,300 naturally occurring beta-lactamases have been defined, and the beta-lactamase database and structure and function, this is a database which is maintained. This particular database shows, as of May 19, 2025, 11,534 beta-lactamases have been reported. The most often the class C beta-lactamases, followed by the class A, okay, and then class D, and class B comes at the end. So let's see how these beta-lactamases will act biochemically. So if you see the beta-lactam ring, okay, and this is a beta-lactamase with its serine molecule at the active catalytic site, and this serine molecule will share an hydrogen ion with the enzyme, and this causes the ion exchange between the enzyme and the water molecule, adjacent water molecule. This adjacent water molecule will cause nucleophile attack on the amine group of this beta-lactam ring, giving rise to the end product and separation of an active enzyme again. So this one particular enzyme can degrade lax together molecule of the beta-lactam antibiotic. This is about the serine beta-lactamases. If you see metallo-beta-lactamases, they have two zinc ion molecules. The two zinc ion molecules, they acquire the charges from where the water molecule will nucleophilically hydrolyze or nucleophile attack of this water molecule to the beta-lactam ring, degrading it to the end product. Majority of the products, they are acidic in nature, penicillin broken down to penicillinoic acid, like that. So similarly, carbapenemases. Yeah. So classification system, if you take, So beta-lactamases are being classified functionally by Bush, Jacobi, Medeiros. This is the oldest classification, and followed by Ambler's classification once the molecular typing started. Somebody is writing, I think. Okay. Yeah. So this is a classification scheme which has been recently published by Karan Bush in 2018 in antimicrobial agents and chemotherapy. So she has listed it in a very beautiful way. So wherein the earlier, if you see the beta-lactamases, initially they used to be characterized based on the serine or metallo-beta-lactamases. Later on, the functional classification came based on the substrate profile and inhibitor profile. And then once the sequencing facilities were available, molecular classification started. And based on the functional molecular classification, the molecular classification has 4 groups, A, B, C, D, and functional group has 1, 2, 3. The molecular class A and D comes under group functional group 2. Molecular class C comes in functional group 1, and molecular class B comes in functional group 3. Okay. So if you see the major subgroups of this A, again, 2A is there, which is a penicillinase, which is inhibited by, so you should see the inhibitor profile also. What is the substrate it acts, and what is the inhibitor it acts, what is the inhibitor which inhibits this enzyme, and the representative genes or the particular beta-lactamases has been listed in the bottom. So 2A, penicillinases. 2B, they have extended towards the first and second generation cephalosporins. Okay, 2BE is the one which is called as extended spectrum beta-lactamases. We also have the 2BR, that means inhibitor resistant beta-lactamases, which will not be inhibited by clavulanic acid, but will be inhibited by avibactam. And this is followed by 2F, which has some of the carbapinamases like KPC and SME. The class C, or the functional class group 1, has 1 and 1E. 1E was called an extended spectrum AMC beta-lactamase, but that has been taken out now. Only one is there, where we have the AMC enzyme, which can be of chromosomal or plasmid mediated. Okay. So and then coming to the class D enzymes, which are oxacillinases, we have, again, depending upon the substrate specificity and inhibitor profile, we have grouped them into 2D, 2DE, and 2F. So we have metalloenzymes, that is class B or group 3, which is having A and B, that I'm going to tell you again. Okay. So just a minute. Can I take out this particular markings? But anyway, somebody has written, it seems. Okay, leave it. So this is just a tabular format, which the Bush Jacobi Medeiros classification, which we made in 2009 for our reference. Then coming to the catalytic site. So generally, most of the enzymes, they have 1 or 2 serine molecules at their active site. Especially this particular thing, that is the serine and followed by 2X and lysine are this is the catalytic site for
I'll
this
finish this
particular
gem extensions and
okay.
okay.
So these are the particular combination of amino acids which is there in the catalytic site of the serine beta-lactamases, whereas metallo-beta-lactamases, they have histidine molecules in their active site, which is tagged with the zinc ions. So this is the class A, B, and D beta-lactamases and their loop structures. These are the beta-pleated loops of L1, L2, and L3, which creates a space through which the beta-lactam antibiotic is going and acting. So that is why the substrate specificity of these beta-lactamases depends upon this particular configuration of the 3 loop structures. And if you see the class D enzymes, class C enzymes, they have another molecule of LC, which is going to reduce the space, and that is the reason these are going to act on cephalosporins and sometimes weak activity on carbaphenemases, but no activity on the penicillins. They are called cephalosporinases. And class D enzymes are oxacillinases, and they have a little bit of elongated space where some of the cephalosporins, penicillins, as well as carbaphenums can bind. This is the group B enzyme. That is, it is divided into group B1A, B1A, B1B, B2, and B3. So most of the enzymes which we see in Indian scenario are NDM, which is seen in B1B group. And we also see some of the IMP and WIM in Pseudomonas and other organisms. So this is the classification scheme, which is given by Karen Bush again, how the beta-lactamases have been classified. So this is little complex to understand, so I have made it a simplified version of the same flowchart. Here you can see there is if you see any reduced microbial or clinical response to any beta-lactam antibiotic in a wild strain, which should be susceptible, then check with a nitrosamine test to check whether it is a beta-lactamase or not. If you find a beta-lactamase, then transfer that particular BLA gene to a suitable gram-negative bacteria, which is sensitive. Okay, so you are going to transfer it just to check whether it is on the mobile genetic element. Okay, or else you can clone it and put it into a bacteria. Okay, then the susceptibility difference between the original and transformant should be made first. Okay, so you are going to see whether there is a difference in the susceptibility between the original, what we isolated in the wild type, and the transformants. And then you are going to check what are the substrates and inhibitors for this particular enzyme. And followed by, you are going to do PCR to say what is the most likely family it belongs to. Then you go for full sequencing, and then later on, to assess the biochemical properties, you just go for protein purification using cloning, and then assess the catalytic properties, hydrolytic properties, the Km values, and other things, and then put it into a functional group as well as molecular group into the database. This is how the beta-lactamases new nomenclature is done. Otherwise, this flow is for only for putting the new beta-lactamase which is invented, not for the routine purposes what we do for detection. Just brief about ESBLs. We know that they have extended the spectrum from first and second penicillin, first and second generation cephalosporins to third generation as well as fourth generation. Okay, so they can hydrolyze most of the organisms except most of the beta-lactams except the carbaphenums and the cephamycins, and they are located on transmissible plasmids and inhibited by clavulanic acid. So here I just want to mention one thing is, most of these beta-lactamases, whatever we are mentioning, they are located either chromosomally, which is intrinsically present in that organism, or due to the flanking regions of IS or the transposons adjacent to the beta-lactamase gene, or by inserting it into a plasmid, the genes have been transferred from one organism to another organism, leading to the selection of these particular strains both in the environment as well as in the humans. So there are a lot of ESBLs. We know that M, SHV, CTXM, all these things, almost all these types of enzymes we have found. Especially more commonly we see TEM, SHV, CTXM, OXA type of enzymes in Indian scenario. So AMC beta-lactamases are called cephalosporinases. These cephalosporinases, one thing you should remember is they have less activity towards the penicillinases and penicillins, and they are not inhibited by clavulanic acid. So if you see the substrate, they have more affinity towards cephamycins like cefoxetine or cefoteton. Okay. So most of the time they can be a chromosomally mediated one, like we can see in Enterobacter cloacae, erogens, Citrobacter, Serratia. So all these things, they have chromosomally mediated AMC beta-lactamases, and these AMC beta-lactamases, they are helpful in the synthesis of cell wall for the bacteria. If you see, there are some AMP-D, AMP-R proteins which regulate the expression of these AMP-C in a routine gram-negative, all these chromosomally mediated AMC beta-lactamases. Generally, AMP-R will go and sit in the repressor site, and it tries to control the AMC production or decrease the AMC production. So once there is a beta-lactam in the periplasmic space, it causes it stops the peptidoglycan synthesis, and thereby the accumulation of the intermediate products will occur, and this accumulation of intermediate products will change the conformation of this AMP R, and AMP R will go and bind to the promoter region instead of the inhibitor repressor region. Once it binds to the promoter region, the excessive production of AMPC will start, and that lead to the constitutive expression of AMPC. So this particular mechanism will be taking place in these organisms. That is why when you are reporting the beta-lactam therapy for these organisms, it is better to ask for a repeat sample after 72 hours, wherein you can expect the expression of AMPC, or else you suggest a alternate antibiotics for this. So this is about the chromosomal AMPC. If you see the plasmid-mediated AMPC, most of the AMPC from this chromosome have been transferred to the plasmid, and these plasmids have been conjugating and transferring the resistance gene to most of the gram-negative bacteria. Now the Klebsiella species or Citrobacter corseri or Proteus mirabilis we see, they don't have any AMPC beta-lactamases per se, but they are carrying this chromosomally mediated AMPC beta-lactamases. But AMPC, as I told, they are resistant to the cefamycins like cefoxitin, but there are certain AMPC which can be sensitive to cefoxitin. I mean, they are getting inhibited by cefoxitin, as well as they will be inhibited by clavulanic acid. So there are some unusual formats of AMPC will be there, and also some of the AMPC like ACC1, they'll be sensitive to the cefoxitin. So cefoxitin, though, because we tend to use it as the marker for AMPC, so it may mislead us. Okay. Then coming to the beta-lactamase inhibitors. If you see the clavulanic acid, which is a traditional beta-lactamase inhibitor, and penicillin, you can see the structure is same. Okay. So I forgot to mention one point is beta-lactam ring will mimic the terminal diala, and wherein the transpeptidases and carboxypeptidases, they get misled and bind to penicillin rather than binding to the PBPs. Okay. So here the clavulanic acid structure and penicillin structure, if you see that the four-membered ring is almost same, and so what happens is the beta-lactamases goes and binds to clavulanic acid instead of penicillin, and penicillin will be spared, which is going to act on the PVPs to inhibit the cell wall. So again, the BLIs have been classified into the classical penicillin-based BLIs like clavulanic acid, tazobactam, or sulbactam, or the first generation non-BLI based non-penicillin based BLIs, that is diazabicyclooctanes derivatives like avibactam, relabactam, and zidibactam have come. And second generation non-penicillin BLIs are boronic acid derivatives like weberbactam or tanirbarbactam or the zerurbactam. Okay, these are the newer molecules which is under phase 2 or phase 3 trials. And choosing the appropriate beta-lactamase inhibitors combination has been done based on various principles. So though it is not relevant here, I'm mentioning it here because few people have the tendency to give unusual combination of beta-lactam and beta-lactam inhibitors combinations, which should not be reported clinically. Okay, so based on various properties like complementary spectrum of activity, whether both of them they have a same kinetics, or whether they are having what is the complication profiles or resistant or the toxicity profiles of individual drugs, and synergistic antibiotic activity, or some of the beta-lactam inhibitors like sulbactam, they themselves they have the antibacterial activity. All those considering all those into consideration, these combinations have been made, and that is why one should know which combination of beta-lactamase inhibitors should be given in the report.
What is that?
So now coming to the various classes of beta-lactamases which are inhibited by various beta-lactamase inhibitors. Okay, so this the list actually it is exhaustive, but I have listed out only few, and these are the beta-lactam inhibitors and the class of antibiotic. The beta-lactamases on which they act. So now we have, because we started exclusively using the BL-BLI combination, so we started getting even resistance to BL-BLI combinations now. That may be because of multiple drug resistance mechanisms. There can be more than one mechanism, or alteration in the amino acid sequences of the PBPs, or decreased in the membrane permeabilities. Okay, that can be one of the possibility. And mutation in the target protein, that is the beta-lactamase mutations, or activation of efflux, that is multiple drug resistance mechanisms. So coming to the other mechanisms other than beta-lactamases, we have the efflux pump, which is the most common family of efflux pump which is involved in the drug resistance, is the resistance nodulation division, that is RND efflux pumps, which is actually functioning as a bacterial physiology, which helps in either extruding the metabolites or for quorum sensing, pathogenesis, or virulence. It's a tripartite protein having inner membrane and the membrane fusion protein and outer porine. And mutation in the regulatory system will lead to overexpression of this efflux pump, leading to the drug resistance. You can see the various the efflux pumps of RND families among different group of bacteria, and for the beta-lactam antibiotics for which they become resistant. Okay, so this is a table which shows that. So if you see, for example, Escherichia coli, ACR-AB, this TOIC, it reduces the susceptibility to penicillin and cephalosporins. Similarly, in case of Pseudomonas acinetobacter, you see, you can see the R&D group of this thing. They cause resistance to penicillin and also some cephalosporins and carbapenems. Okay, and for Campylobacter, Neisseria, and Klebsiella, these are the R&D families which leads to the reduced susceptibility to penicillins. The another mechanism what the organisms have developed for the development of resistance to beta-lactams is oftenly seen also. So it is not often present as a sole mechanism. Sometimes it can be in combination with a beta-lactam or a flux pump or so and so. Okay, so if you see, OMP-F and OMP-C are the major porine channels which is present in E. coli. In the presence of the hyperproduction of the beta-lactamases, they may cause resistance to penicillin, cephalosporin, carbapenems, and monobactams. Likely, the Klebsiella, OMP35 and 36, Enterobacter, OMP35 and 36, Pseudomonas, OPRD, Acinetobacter, this CAR-O or OMP-A have been associated with the porine loss, and that leads to the corresponding resistance to few of the beta-lactam antibiotics. So the combined mechanism has become a headache now. So wherein there will be an efflux with a beta-lactamase or a porine loss with a beta-lactamase, and that particular phenotypes are difficult to identify unless we go for specific detection methods. So now after all these things, I will go to the resistance detection methods. Those who are unmuted, please mute yourself. Something like Anita, JP, Sangamitra, Rameti, they're all, it disturbs us actually. Can you please mute it? Yeah. So let's start with the ESPL detection. So if traditionally we used to detect the ESPLs by double disc diffusion test when we were not having the ceftazidime clavulanic acid or cefataxime clavulanic acid. So there we used to keep amoxicillin clavulanic acid in the center, place the 5 different cephalosporins around it to detect the ESPLs. Okay, so but again, the problem was they used to recommend initially 30mm gap between the edge to edge. Later on, if it is not showing proper result, then you reduce it to 20 or 30. That particular alterations were required, and it used to take 2 to 3 days for interpreting, sometimes for few isolates. Okay, so you can see a keyhole pattern of extension towards the clavulanic acid, which means that there is a possibility of a beta-lactamase in this particular organism. But the problem is again to be ER enzymes, which will not be detected by this method, and this is a less sensitive method which is not used nowadays. So then people came up with 3-dimensional test. This 3-dimensional test means you are going to grow the bacteria for some time into a log phase, pellet it, and then add a suitable lysis buffer, or you put it into a liquid nitrogen or into a freezer, -80 freezer. Do 5 to 6 cycles of freeze and thaw so that the protein is retained, but the cell wall is broken, because most of the beta-lactamases, if you see in gram-negative bacteria, they are in the periplasmic space. So you have to break down the cell wall to release it into the environment, I mean, the medium. So then you are going to punch a hole in the media and cut a line just perpendicular to it, lawn the ATCC E. coli, and put the either cefoxtin or ceftazidime, whatever the antibiotic you want, and load your the protein extract into this well. And if you see any indentation in the zone of inhibition, then it says that the particular organism produces a beta-lactamase. But this is again a cumbersome test which is not followed nowadays. Disc replacement method was also introduced, not much useful. And then agar supplemented with clavulanic acid was tried. It is successful. Definitely it is good, but clavulanic acid is a very costly drug. If you see 50 milligram costs around 1.5 to 2 lakh rupees. And then coming to the last method, now the CLSA recommended method, wherein we can say it's a phenotypic confirmation method for detection of ESBLs, that is using cephalosporin and cephalosporin clavulanic acid combinations. And if there is an increase in more than 5 millimeter diameter between the drug with and without BLI, we can interpret as extended spectrum beta-lactamases. So chromogenic cephalosporins like nitrosamine can be used, but we cannot differentiate whether it's an ESBL or AMC, because it can, any beta-lactamase can hydrolyze this and change the color from yellow to red. So there are some iodometric methods, that is in the presence of iodine and starch, the iodine will be reduced, and that causes the starch to turn from black to the colorless. And this method is also was widely used earlier but not used nowadays. And acidometric method, wherein we use the acid base indicators like either phenol red or the various other acid base indicators can be used. Phenol red is most oftenly used, and we can see the change in color because the end product of the beta-lactam hydrolysis of any of the beta-lactamases is acidic in nature. It changes the color of the antibiotic solution from pink to yellow. So molecular techniques like DNA probe, PCR, RFLP, multiplex PCR, real-time PCR, definitely they are in use, and they are now considered almost like a gold standard, because you can detect the gene particularly, but they do have some of the limitations which I'll be telling in the subsequent slides. So again, the diagnostic problems are also there with the ESBL detection. There is no simple marker. Like one marker cannot be used because the different ESBLs have different substrate preferences, so which makes them difficult to interpret by using only ceftazidime clavulanic acid, or cefoxam clavulanic acid. Sometimes we may have to use multiple combinations of different cephalosporins. That 5 indicator screening cephalosporins which was suggested in CLSI may be used to detect. And also ESBL producers may harbor multiple ESBLs, or there can be an AMC associated with, which may intervene with that interpretation of the phenotypic confirmation test. And also inoculum effect. That means as you go on increasing the inoculum concentration of ESBL producing organism, the drug resistance also increases. That inoculum effect is seen both in vitro as well as in vivo, especially the mucosal associated infections of ESBL producing organisms like UTI or endocarditis. They're often associated with this inoculum effect, where the bacterial concentration is more, there the resistance is more. Coming to detection of the AMC beta-lactamases. So here, this is, though we know that the cefoxtin is a substrate for AMC beta-lactamases that can be used for a screening of AMC. But the problem is, as I told earlier, some of the AMC beta-lactamases may be sensitive to cefoxitin, like ACC, and this may show false susceptibility. Maybe you are interpreting as AMC negative, or there can be a possibility of porin loss. Okay, porin loss lead to cefoxitin resistance, and that is going to cause again maybe interpreted as AMC. Okay, so for that people came out with cloxacillin. Cloxacillin is going to inhibit few AMC enzymes. So cefoxtin with cloxacillin has been tried with success, but it may fail in some of the AMC beta-lactamases. And this is particular PBA, that is phenylboronic acid. again, it is a very good molecule which can be used in combination with cefoxytin as a disk potentiation test, and this can confirm the AMC production. But again, the problem is the same phenyl boronic acid can inhibit the carbapenemase, which is also acting on cefoxytin, and you may misidentify even a carbapenemase as AMC. Though majority of the time, what we are seeing is an AMC will be there even with carbapenemase also. Okay. And the three-dimensional extract test. This is a good method. Again, we cannot differentiate because substrate profile of carbapenemase and AMC is matching. In the presence of carbapenemase or AMC, this particular three-dimensional test can give a positive result. And people came out with a simplified version of three-dimensional test, which is called as the AMC disk test. So we have also tried this particular test, but it all depends on technique. And again, it is an indirect method because we are putting the organism onto the disk, and we are learning the ATCC E. coli, and then we are putting cefoxytin disk. And adjacent to this, we are keeping so that the enzyme produced by the test train is going to hydrolyze the cefoxytin, and that gives rise to a cloverleaf pattern or indentation to the zone of inhibition. And AMC induction test, as I told, inducible AMC can be detected with the help of an inducers. Inducers for this inducible AMC can be like beta-lactamase inhibitors like clavulanic acid. Very good inducer is imipenem, and even sometimes cefoxytin. Cefoxytin is also a very good inducer of AMC. So this is a combination method of clavulanic acid and boronic acid using cefotoxime and cefepime, which we published in 2011, wherein we were able to differentiate the ESBL and AMC in the whether they are alone or in combination, and this can even differentiate the carbapenemases also. Coming to detection of the carbapenemases, the carbapenemase detection, again, it started from the beginning itself, where they started with for detecting the metallobeta-lactamases especially. So, excuse me. So initially, the SME type of metallobeta-lactamases which were detected, they were identified based on using EDTA. So already this potentiation for detection of carbapenemase was well identified in 1980s itself. But later on, CLSI approved certain methods to identify the especially the carbapenemases. One of which was this modified Hodge test. Modified Hodge test because it was less sensitive for detection, and it was dropped down from CLSI, and now it is not been recommended by CLSI. So these are some of the other old tests which I'm just list out and go to the proper test. So boronic acid can be used for disk potentiation to detect the serine carbapenemases, or EDTA can be used, or this dipicolonic acid or dipicolinic acid can be used for inhibition of this MBLs. Okay, so again, inclusion of the dipicolinic acid, EDT or EDTA with boronic acid, with cloxacillin, with clavulanic acid, different combinations have been made. If you remember, Dr. Sumit will be telling about different combinations of beta-lactamase inhibitors to identify the single or multiple beta-lactamases in a particular strain. So this is imipenem EDTA method. So wherein we can differentiate the metallobeta-lactamase. So we are going to add 0.5 millimolar concentration of EDTA to the disk and place it onto the surface. So there are colorimetric methods. Say, for example, I told when the beta-lactam ring is broken down, it leads to an acidic end product, and this acidic end product can change the color of some of the indicators like phenol red or bromocresol purple or cresol red, all those things. And based on that, Nordman and Pyrrell in 2012, they introduced a test called Carba-NP test. And after that, a lot of versions came like Blue Carba, Rapid Carba, Rasco Rapid Carba screening, all those commercial screening tests came. These results are you are going to get within 2.5 hours. sometimes what we have observed is even within 10 minutes, the results will come. And you can, especially with respect to MBLs, the method is highly sensitive. Maybe we are facing some issues with this method for OXA type of enzymes, which is OXA 48 like especially. So they are poor or weak hydrolyzers, which will not give rise to sufficient acidity for color change of the indicator. So based on this, we published 2 papers wherein we just modified this carba-NP test, and we added a third tube wherein we included EDTA. So if it is a serine beta-lactamase, the third tube will remain yellow, and if it is a metallo-beta-lactamase, the third tube will get inhibited, and it becomes pink. It remains pink. So by this way, we can differentiate the serine and metallo-beta-lactamases with this particular method. So this is a published method in JCDR, I think 2021 we published, and you can go through the article. So now currently the CLSI is recommending to go do modified carbapenum inactivation method. It is called MCIM, or also to detect to differentiate the serine from metallo-beta-lactamases. ECIM is also been indicated. So so here the suspected organism will be grown in tryptic soy broth, and an imipenem disc will be added and incubated for 4 hours. So later on, ATCC E. coli will be launched on the Muller and agar, and you are going to place the particular disc from that broth, and you are going to place it on the media. If you see a zone of inhibition of more than 19 millimeter, it is considered carbapenem is negative. If it is around 18, it is inconclusive, and if it is below 18, it is considered as positive for carbapenemases. And if you add on EDTA in a concentration of 5 millimoles per ml, so it can inhibit the MBLs if it is there, and thereby the increase in the zone by more than 5 millimeter between the plain and EDTA added imipenem tube, we can differentiate the MBLs also. That is called MCIM and ECIM method, which is claimed to be 98% sensitivity for detection of most of the carbapenemases, whether it is class A or class B. Class A, D, or B, anything. Okay, but I'm not in favor of this particular test because it takes too much of time. But if sufficient time is available for detection, then of course this is good method for detection. Then a lot of gradient methods have come out, wherein the E strips, which are been coated with the combination of drugs. This is, for example, cefotoxime, clavulanic acid, or imipenem, EDTA. Okay, so you can see the change in the MIC, that is 4-fold decrease in MIC between the plain and EDTA associated strips, and you can interpret as either ESBL, carbapenemase, or so on. So as we are seeing the serine and metallo-beta-lactamases, the people have already published using ceftazidime, avibactam, and astronome combinations, okay, to interpret whether it is pure MBL producer or pure serine carbapenemase producer, or there is a combination of both MBL and serine carbapenemases. And then coming to if you want to look for any synergistic activities, checkerboard MICs are recommended, where we are doing a different combination of different antibiotics, and to look for the synergistic activity of the various molecules. Okay. Then chromogenic agar method, they are actually selective medias to isolate from environmental samples as well as contaminated specimens. The drug resistance as well as species can be identified with this, and they are helpful for only screening and as a part of infection control. Though they reduce the turnaround time, the cost, and also the further confirmation which is required will limit their use, but still they are good screening media if you can include in your diagnostics. Then Malditoff based methods have been now coming popular, because there is a very short turnaround time of 30 minutes or up to 2 to 4 hours, depending upon what method you are adopting. If the library is having the mass spec peaks of the particular beta-lactamase, you can just load the organism and see the peaks, or you can use indirect methods. For example, you can incubate imipenem with a particular organism first you spike it into the MALDI and look for the peak. And then after 1 hour or after 4 hours, you are going to check for the peaks. If there is a reduction in the imipenem peak, then it indicates that there is a carbapenemase which is hydrolyzing the imipenem. Similarly, if you change the substrate, you can detect that particular enzyme also. Immunochromatographic card tests are available, wherein the 5 carbapenemases already there. It is in the market. Even some of the ESPLs can also be detected by lateral flow assay. So Coris is a company which manufactured it earlier. So this is going to detect from the pure cultures of the growth from the plate, and it can detect within 5 to 10 minutes. Spectrophotometric methods are being used since many years. These methods can again be used for the detection of carbapenemases, and then it can be done within 40 minutes sometimes. Okay, so here the spectrum of the lysis of the imipenem or a particular substrate is studied over a period of 40 minutes at a particular wavelength, and the spectra is drawn and to see the hydrolysis profiles. This is how the kinetics also been studied for this beta-lactamases. And bioluminescence is also used for the detection of the carbapenemases. And electrochemical methods also been introduced, like BYG carba test, wherein the sensor detects the potential change between the electrodes when the carbapenemase is hydrolyzing the beta-lactam antibiotics, and it can detect as early as 30 minutes with a real-time curve. This is a prototype which was designed in 2016, and this is an excellent one which uses the Arduino Uno board to detect the carbapenemases. So definitely this type of devices will come in future for detecting the beta-lactamases in the laboratory. Automation, definitely the advanced expert system will predict the AMR, and sometimes it confirms also. But again, there may be some error rates, and some of the phenotypes may be difficult to detect with automated systems. And molecular methods are still considered as gold standard, because the turnover time you can detect in the same day directly from the clinical specimen or from the pure culture. Okay, so and also because the option of multiplexing is there, so we can go multiplexing of conventional up to 6, or even if it is real time, we can do multiplexing to 5, depending upon the machine which you use. So this is a multiplex PCR which we generally perform in our laboratory. So where it detects the 5 different carbapenemases, NDM, OXA, and VIM. You can see some of the organisms, they possess both NDM and OXA together, okay, which makes the situation more complicated. And this is a table. I think I'll not go into the deep of this particular table as the time is limited. So you can go through the CLSI guidelines of table G3, where they have clearly given how to use the molecular test in combination with the phenotype to interpret and guide the clinician for appropriate therapy. So you can go through these tables. So because of time factors, I'll not be explaining in detail about this. So you can refer CLSI document for this. Then based on the molecular methods itself, the gene expert and other automated systems have also come up. And the newer technologies, not the new, we can say this is some technologies like isothermal amplification also been tried with either LAMP or the RPA based test. So this is a loop-weighted isothermal amplification assay which we developed for NDM. So it is a colorimetric. With a naked eye, you can see this is a negative one, and all these are positive at various concentrations of DNA. But the thing is, it is prone, because it's high sensitivity, it is prone to a lot of non-specific amplification and contamination that we are not recommending. So microarray is one of the method of detection of the carbapenemases. Here, the oligonucleotide sequences will be immobilized on multiple points, and it can simultaneously detect antibiotic resistant genes as well as the particular bacteria, and even some of the virulence genes also. And it is good. Like before the introduction of NGS or WGS, microarray based methods were frequently used. Still some of the microarray based methods are there, like BioFair film array or virgin system or checkpoints. So these are the things which are available. And then now the era of next generation sequencing, we can go for whole genome sequencing using either short read or long read sequencers like Illumina or the Ion Torrent or the ONT platforms. And especially the ONT platforms, they can give real-time detection of the drug resistance while doing the sequencing. And also there are certain pipelines. Once the WGS is done, we can put the raw files or the refined files into the bioinformatic tools, and the pipelines will take you, and it will clearly depict all the resistance mechanisms. And also manually you can use the Linux based systems, or you can use the online platforms of ResFinder or the card or pathogen watch. You can upload your sequences and get all those mobile genetic elements as well as the different beta-lactamases present in the bacteria. So this is the most reliable method, but the high cost, turnaround time, and difficult for data management. And also the analysis part is also not so easy. So this is limiting, but still one day everything gets automated, and it will become a routine practice in our future. This is just a pipeline, like, or we can say algorithm for which the recently published molecular approach towards identification of drug resistance. And lastly, the limitation of molecular test is the negative test does not imply that the resistance is not there, because novel mechanisms may not be targeted by this unless it is an WGS or NGS. And the presence of gene does not imply the expression. Again, that is the thing which is given in that CLSA table. Okay, the 3G. And you have to remember that expression is also important. Expression, the mere presence of gene will not indicate that it's a resistant by phenotype. And unidentified or novel variants cannot be detected, and it can overestimate the antibiotic resistance. And testing on primary samples, okay, especially as contaminated specimens, it is very difficult to say whether the antibiotic resistance we are detecting by molecular, is it truly from the pathogen which we are isolating, or is a bystander which may be there, or a mixture of growth. Okay. Then coming to the last slides, where we are detecting the other mechanisms. We can detect the efflux pump using efflux pump inhibitors. For example, we can use the phenylalanine, arginine, naphthalamide dihydrochloride, PABN, which is called, which inhibits the R&D family of the efflux pumps. And also the CCCP, the carbonyl cyanide chlorophenyl hydrazine. It is again a very good molecule which we have tested. Both we have tested the PABN as well as CCCP, and they work very well for the efflux pump inhibition. And if you want to completely identify the beta-lactam resistance, you have to use all modalities to come up with the identification. And also 2,4-dinitrophenol, they're all efflux pump inhibitors. So here again, 2,4 declares in the MIC, we can identify there is an efflux pump overexpression. Loss of porins can be detected by using SDS page. So they are going to extract the cell wall proteins and do a high speed centrifugation, 1 lakh RPM, and the particular portion, a layer of the layers will be taken and run in the SDS page to look for loss of a particular protein. So you can see OMP 36 and 35 are lost in this particular strain compared to 86C. And this is just the approach towards the phenotypic, molecular, and as well as the mass spec based methods of detection of AMR. And this, I am going to conclude the presentation. And if there is any doubts, the discussion is open.
That was an amazing presentation, Dr. Rudresh. You covered beta-lactams from like A to Z, starting with their structure, classification, then delving into resistance mechanisms and details of testing the resistance in bacteria. Your coverage of both phenotypic methods and newer proteomic-based electrochemical approaches was really brilliant. Thank you so much for such a comprehensive and insightful session. This
Thank you,
topic
sir.
itself is so vast, and I should congratulate you for beautifully managing the time and bringing in so many aspects about this topic in one go. Okay, there are a lot of questions being typed.
Yeah. I will see the
Yeah,
chat list, sir.
one question is by
Not
Dr.
clear.
Sneha Madam, Sneha Chinchillur Madam. What
Ah,
is what about yes SIM? We know M SIM, E SIM.
uh-huh. Yes, Sim, sir.
Yeah.
I have not heard.
Okay.
Maybe she, maybe I had to see, sir. Maybe I have not heard about this Sim.
Okay. All right.
Yeah.
Then how reliable are rapid card tests for detecting resistance?
Especially lateral flow assays have been found very good in identifying the carbapanimases. They are time tested and they are working. Only thing is the cost. Each card costs almost 1,500 to 2,000 rupees. I think local companies have come out with detection of commonly occurring beta-lactamases like NDM, OXA, with some of the TEM and other ESBLs. I feel they should be the point of care diagnostics which should be used by us if any critical cases is there in the clinics.
Okay. So one question is how effective is a disk approximation test to detect inducible AMC beta lactamase?
Of course, that is the best method. You can detect only by placing an inducer near around 15 to 20 millimeters,
Yes.
especially imipenem. So we tend to put imipenem in the center and put all the cephalosporins around it so that the induction, that flattening of zone can be seen easily.
Okay. In fact, I actually imitated one of your publications long back, that is combined method of assessing, detecting multiple resistance in a single plane.
Yes,
That was very good. Uh, I think they have a lot of questions, but
yes, sir.
they are still typing. So meanwhile, let me ask a general question. So
Yes, sir.
what are your suggestions for postgraduate students to master the concepts about this resistance and the detection and keep themselves updated?
Sir, mainly follow those publications who are experts in the field because that is what we do. Generally, I follow Karen Bush always. Wherever she publishes, I never miss a single publication.
Okay.
If it is not accessible, I'll write to her, get her article, and read. So that urge should be there. Other thing is basically you have to understand the principle on which the beta lactamases act or any particular drug resistance mechanism acts. So if you understand the core mechanism, rest all becomes very easy. I think it was Mandel which opened our eyes towards Bush-Jacobi Medrose in 2008.
Mhm.
So when we read Mandel, we understood that there is something called ESBL, AMPC. Then I think that arose a lot of interest for me in detecting these type of drug resistance. And as a first year PG itself, I started working, especially in 2008-9 itself, we started doing three-dimensional tests. Just do the regular refrigerator, just freeze thaw and centrifuge use the supernatant for doing three-dimensional, and it excellent actually. And
Amazing.
also trying it hands-on will give you a lot of confidence. Second thing is, see, approach. That is more important. See, 2009, I went 70 kilometers in my three-wheeler to get clavulanic acid from Medrich Pharmaceuticals. So my guide Nagaratnama madam, she scolded. You went around 12 o'clock in the night, you are coming back. What if something would have happened? So that type of urge should be there from the postgraduate students. You see, clavulanic acid, if you say, I told 150 milligram is around 2 lakhs. And we got 1 gram from Medrich Pharmaceuticals. And it
Oh,
lasted for almost 4 years. And we did extensive work on beta lactamases at that time.
wonderful. Uh, I'm very sure a lot of them are currently working on some research topic, either it is because of their thesis or otherwise, related to beta lactams, their resistance mechanisms, all that. So, considering so many new things have come up, and also
Yes.
considering that they have limited time and limited budget, can you suggest some topics related to this topic?
Sir, actually, now uh, if you see certain institutions, they do even whole genome sequencing free of cost.
Mhm.
Okay, so there is no need for you to invest anything. And even if you invest also, it's very meager amount. For example, National Antibiotic Resistance, that manual which is released in 2022, it has every aspect of beta-lactam resistance detection, including molecular. They have given exactly what is the sequence, what is the amplicon, how to prepare gel, everything has been given. Okay. So, and the cost of primers and other things, we should come out of that concept of PCR is very costly. So hardly I can say to do in my lab, we do PCR in and out. Maybe I am wasting PCR reagents of 30 to 40 reactions as it is daily. Okay. So we don't mind because the cost is so low now. I'll get 1,000 rupees 100 test of PCR, and primers are very cheap. One set of primer is 800 rupees. So they can definitely go with the multiplex PCR because after the COVID-19, all of us have real-time PCRs at least with us, and it is not. There is no thing like you need to use only conventional PCR for doing conventional PCR. You can use real-time PCR also for doing conventional PCR.
Fine.
Okay, just you need to switch off that
Okay.
camera capture. That's all. You can use your real-time PCR for as a conventional PCR also. Use it. Okay,
Fantastic.
and extraction. When come to extraction, see the cost of this any detection. You can use Carba-NP. Carba-NP is a very simple test which not even cost 25 paise. If you purchase 500 rupees phenol red and imipenem cilastatin 400 rupees, you can do thousands of tests. In ESIC, in my previous institution, I have done almost 2,000, 3,000 Carba-NP tests. And it was a test which is like our indole and IMIC reaction. We used to do it as a routine,
Yes.
Carba-NP. That is one thing. Second thing is molecular, sir. Coming to molecular, now the molecular is most important for any postgraduate to do. Because see, if you go to any research institutions, all the AMR related works are taken up by the basic scientists who don't have the clinical knowledge. That is a major problem actually. Whoever is getting the fund is they are in the basic institutions where they beg us for the drug resistance trials. Okay, but we have treasure in our hand, but
Yes.
we are not knowing it. Eyes cannot see what mind doesn't know. So we have a
Yes.
treasure. Okay, so you use optimally these technologies and exploit it. See, hardly the entire carbapenemase set of primers costs somewhere around 5,000 rupees. And if you are testing around, say, your sample size in dissertation may not cross 300 to 400, order some 3-4 master mix, which is cost around 4,000 rupees. In 10,000 rupees, you are going to do everything. Only thing is, you must have gel electrophoresis, the agarose, TAE buffer, all those things are required. Of course, I'm not denying that. The other thing is extraction part. See, there are simple techniques to extract. Just put your organism at 0.5 microgram concentration in a say 200 microliter of your saline. Heat it at 95 degrees Celsius in a PCR thermocycler itself. Keep one cycle of 95 degrees Celsius for 5 minutes. Take it out. Just use 2 microliter out of it as a template, DNA template. Your PCR comes very nicely. That's called colony PCR. Okay, there are some shortcut methods which gives very good shortcuts to cut down your monetary part and do it. And of course, say now sometimes if, for example, when I was doing PG, we were struggling to get the molecules. Okay, for example, cephatoxime, ceftazidime. Now also, I don't purchase imipenem pure powder for doing Carba-NP. I use imipenem cilastatin, which is available commercially, to do my Carba-NP test, and it gives very good result. Today only I showed my PGs about the Carba-NP test.
Amazing. So it is
Yes, sir.
evident how passionate you are about this topic and how much in depth you have worked and understood. We are actually fortunate to have you uh, to cover this topic in master class. I think there are a lot more questions, and because the topic is so vast and a little complex, many people might come up with questions later also. In considering
Sir,
all that, we have
they
generated.
can ping me. They can ping me
Okay,
anytime to my mail ID, and
yeah,
I'm leaving my mail ID in the chat box. And anytime they can ping me, I can help them for because whenever I'm free, I'm definitely going to reply for that.
wonderful. Thank you so much for sharing. And we also have a link for feedback and suggestions. Participants can leave their questions there also. We will try to compile answers for them and share in the next masterclass. That is another way how it can be done.
Yes.
Okay. I have a couple of announcements to do. Our next masterclass is scheduled for August 8th. The topic is antibiotic susceptibility testing, conventional and automated methods. Actually, there was a request to cover this one first, but there was a little gap in planning, so it's okay. Anyway, recordings are available. People can watch anytime. And the topic will be covered by Dr. Archana Bhatt, uh, Associate Professor, KMC Mangaluru. Uh, Also, don't miss the first quarterly CME of IMMKC on a highly relevant topic. The topic is data to decision, AI in antibiotic policy and prescription. So this is being organized by Microbiology Department at SDM College of Medical Sciences, Dharwad, on August 2nd. Good thing is it is in hybrid mode, and the last date for registration is July 30th. The registration link I will share in the chat box. I will keep this webinar for a longer time so that people can copy all these useful links. With this, shall we conclude? Already it is 7:18.
Good evening, Dr. Rudresh sir.
Yes, madam.
Yeah, we would like
Good
to
evening, ma'am.
thank
Good
on
evening.
behalf of IMMKC. It was an extensive lecture, especially the PPTs were so beautiful and all those tests and all you have put the pics. It will be so helpful for the PGs and even for us also. It was very nice to see the PPTs and your lecture was extensively good, sir. And I
Thank you,
thank
ma'am.
you
Thank you.
for taking out the time for making this lecture.
Thank you, ma'am. Thank you.
Thank
Thank
you.
you. Thank
Thank
you.
you so much.
Just before we wrap up, I want to extend my heartfelt thanks to Dr. Rudresh for generously sharing his immense knowledge and expertise with us today. We truly hope to have you again for future sessions.
Definitely, sir.
My sincere gratitude also goes to the Masterclass Committee and the office bearers of IMM Karnataka chapter for this incredible opportunity, and a special thanks to Dr. Sneha Chunchanur Madam, Dr. Satyanarayana Sir, Dr. Kaleem Bahadur and Dr. Muhammad Iqbal for their invaluable help and support in organizing today's masterclass. So with this, let us conclude today's meeting. The meeting as such uh, will be running for another 5-10 minutes so that people can copy the links. So whoever wants to log out of the meeting can log out. Thank you so much. Thank you everyone for joining.
Bye, sir.
Bye, Dr. Drish. Thank you.
Thank you, sir. Thank you.
Bye, everyone.
Bye, everyone.